Hello, my name is Lorraine Abonono, and I performed my research study at UT Arlington this summer, and my project was over the effects of oxidative stress on mesenchymal stem cells. To give you a little background on my subject, traumatic injuries, especially bone traumatic injuries, are generally associated with an accumulation of reactive oxygen species, which are also known as ROS. This ROS causes oxidative stress at the site of injury, which leads to the degeneration of the cells and hinders their growth. We use mesenchymal stem cells because they are multipotent adult stem cells that can differentiate into bone cells. In our study, we induced oxidative stress with hydrogen peroxide, which has been used in prior studies and proven to be a pro-oxidant. We aimed to determine the smallest dose of the hydrogen peroxide that could have a significant impact on the cells. To accomplish this aim, we started by preparing different concentrations of hydrogen peroxide that ranged from 0.2 to 0.8 millimolars. Then we grew our cells in basal media on a tissue cell plate for 24 hours. After 24 hours, we introduced hydrogen peroxide, and after another 24 hours, we performed two viability tests on the cells that had grown. The first one was live dead assay. It uses two dyes, ethidium homodimer and calcine. Ethidium homodimer gives a red color to the dead cells, and calcine gives a green color to the live cells. We took fluorescent pictures of our cells, which can be seen on the top right of the screen. At 0 millimolars, there was no hydrogen peroxide in the cells, and we can see that there was a lot of growth. However, as the concentration of hydrogen peroxide increases, there is a reduction in the growth of the cells, and at 0.8 millimolars, all the cells died. The second test that we performed was MTS assay, which uses an MTS solution, and when that solution is added onto the cells, they give a formazine, which is a purple color. And we use a microplate reader, in this case SpectrumX i3X, to quantify the intensity of the color. We graphed our data, which gave us this beautiful graph in the center. And we can see from the graph that at 0.2 molars, the cells died or started to die. However, the first significant impact that we had was at 0.4 millimolars. And then at 0.8, also supported by this graph, all the cells died. We decided to observe the cells from the 0.4 millimolars group after 3 days. We removed the hydrogen peroxide, and despite that removal, we can see from this picture at the bottom right, that the cells were continuing to die despite no oxidative stress being induced, which shows that they remembered the stress that was induced before. This can be used in future studies as well as the concentration 0.4 millimolars to test the efficacy of antioxidant coatings. These coatings will be used to reverse what ROS does on the cells, and so they will be promoting growth in the cells instead. I would like to thank LSAMP and Dr. Varanasi for giving me the opportunity to perform and be a part of this project. Thank you to Neela Mahuja and Kamal Awad for being my guides throughout the summer. Thank you to Ayat Omar and Shambhavi Anand for supporting me throughout this project. Thank you very much.